Contrary to what you might have seen in the movies, cloning is not about making copies of people or animals, but rather it's about making recombinant DNA that's able to self-perpetuate. So let's say you have some DNA, but there's one part that you're interested in and you want to either store it or make copies of it or just maintain it for future use. What you'll need to do is create a piece of DNA, usually circular DNA or a plasmid, which contains your DNA of interest, which you can put into a living organism such as a bacterium, and it will replicate as the organism replicates. For this to work, you need to have some other things in your plasmid. So first of all, it needs to have an origin of replication, since otherwise it won't be able to replicate when the cell divides. What else do we need in the plasmid? Well, one thing we have to be concerned about is that some bacteria might lose the plasmid, and over time those bacteria might take over the culture. So we want to make sure that only bacteria with our plasmid are able to proliferate. We can do this by adding an antibiotic resistance gene to our plasmid, and then we'll only grow the bacteria in media that has an antibiotic. So any bacteria without our plasmid will die. So how do we put this all together? The first step is to isolate the DNA you're trying to clone. One way to do this is by cutting the DNA upstream and downstream from the site you're interested in, using enzymes called restriction enzymes, which can cut the DNA at specific sequences. If the DNA you're interested in happens to be a gene, you can also collect the RNA you're interested in and reverse transcribe it to make the corresponding cDNA. So what's the difference between a cDNA and the original DNA sequence? Since cDNA is made from mRNA, it has no introns. So now we have our DNA of interest. Next, we need a plasmid. Fortunately, we're in the 21st century, and you can just buy a plasmid that already has everything you want in it, like antibiotic resistance regions and the origin of replication, and a bunch of different sites that are targeted by different restriction enzymes, which you can use to cut open the plasmid and insert your sequence. As a flashback to our blotting procedures a few slides ago, do you remember which blotting procedure would allow you to determine which DNA sequence or restriction enzyme can bind to? That would be the southwestern blot, which is used to detect DNA binding proteins. So first, you'll use a restriction enzyme to cut open your plasmid, and then add your DNA of interest to the mix. And what enzyme do you think we can use to seal them together? This would be ligase, just like in regular DNA replication. Then you can use a process called transformation to get this plasmid into a bacteria and then let them divide. So what's the point of cloning? Well, one purpose is to amplify DNA, just like in PCR, which I talked about earlier. So if you clone DNA into bacteria and then let them do what they do best, which is divide, you end up with a lot more copies of DNA than what you started with. Another use is to make a library of all the genes which are expressed in your sample, which is called a cDNA library. In this case, you're not cloning a specific sequence, but instead you're reverse transcribing all of the mRNAs that are present into DNA, and then each bacterium will get one, so your resulting population of bacteria will contain a library of DNA corresponding to all the genes that were expressed. Let's do one more flash quiz slide for this section. So in addition to your DNA of interest, what two features should be on a plasmid you use to clone DNA? This would be an origin of replication, since without it the plasmid won't replicate along with the rest of the DNA in the cell, and an antibiotic resistance gene so that you can select for cells that have your plasmid in them. This slide is more relevant to research than clinical practice, but these are still concepts that are good to be familiar with. So here's a question that researchers are investigating all the time. What does a particular gene, which I'll call gene X, do? Or usually the same question is, what is the function of the protein encoded by this gene? And I'm showing gene X as the red spot in these homologous chromosomes. A professor of mine used to use an analogy that to figure out what a window in a house is for, you should throw a rock through it, and then you'll figure out pretty quickly what it does. You can't maintain the temperature of your house, and water gets in when it rains. So these are probably the functions of the window. This is usually a good place to start when investigating what a gene does. Get rid of it and see how it affects the cell or the whole organism. You can either knock out one copy of a gene to see the effects of reduced expression, or knock out both copies and see what happens if you completely are deficient in that gene. A lot of the time, knocking a gene out entirely will be lethal, which prevents you from studying it, so you have to look at a single knockout. The opposite approach would be to add extra copies of the gene and see what that does. There are a few strategies for manipulating gene expression in animal models. A knockout animal is one in which a particular gene has been removed, and similarly, a knock-in animal is one in which a gene has been added. One way to knock a gene in is to insert the gene randomly, in which case you don't know where it's going. In fact, it could end up in the middle of another gene, which would disrupt or change its function. A safer method is called homologous recombination, which is used to target the gene to a specific location. To use homologous recombination, you first have to know where you want to put the gene. So let's say the target region of your genome is represented by these letters, and you want to put the gene right here, between the C and the G. By flanking your gene with sequences on either side of it which are complementary to where you want to insert it, it will naturally localize to this exact spot. That's the homologous part. Then recombination can occur in the same way it does during normal meiosis, and you end up with your gene inserted right where you want it. The Cree-lock system is a method that can be used to knock a gene out, and it's controllable so you can knock it out at a specific time. This is useful because sometimes you want to see what the effect of removing a gene from an adult would be, even though you know that removing it from an embryo would be lethal. So to set up this system, you start with two different mice. In one of them you insert the gene Cree downstream from an inducible promoter which you can activate whenever you want, such as by adding a drug. So Cree won't be expressed at all until you add the drug. In the second mouse, you will have your gene of interest with a sequence called LOXP on either side of it. When you mate these mice together, some offspring will receive both the Cree construct and the LOX construct. When Cree is expressed, it produces an enzyme that causes recombination between LOXP sites. This makes them form a loop, and anything that was between them gets removed. So this will knock out your target gene. The last method of gene expression I'll cover is called RNA interference, or RNAi. This method targets mRNA rather than DNA to block gene expression. This is actually a really complex process with a lot of steps and a lot of different variations, but fortunately, you only need the basics for the step 1. So basically, you make double-stranded RNA which is complementary to the mRNA you're trying to block, and transfect this double-stranded RNA into cells. It will stick to its complementary mRNA, and cells will recognize this as unnatural and possibly dangerous, and will degrade all the RNA that's involved, so it won't be translated. Alright, let's finish off lab techniques with a board-style question. So a 40-year-old woman presents to a local clinic because she is concerned that she may have contracted HIV from a former lover. Blood is drawn and sent for testing. In the molecular technique used as an initial screening test to determine if a person has been exposed to HIV, which process takes place if the test is positive? So as usual, go ahead and pause the video for a minute if you want to think about this. Okay, so there are two types of tests that are used for HIV, which are Western blots and ELISAs. One of them is more sensitive and therefore is used more for screening, and the other is more specific and is used to confirm the diagnosis. Do you remember which is which? So the ELISA is more sensitive, so it's used as the initial screening test, which we're going to use here, and the Western is more specific since it also accounts for the size of the protein, so that would be used later to confirm the test. The process from this list that takes place in an ELISA is B, the antigen-antibody reaction. Now some of you may have picked A, and you can actually make a case for A since maybe the antigen is a specific protein, but this is one of those examples where you need to pick the best answer, not one that's arguably correct. Unfortunately, some step 1 questions will try to get you like this, so always read all the answer choices even if you see one that seems good. While we're here, let's go through the other answer choices. So A would be a Western blot, since you hybridize antibodies to your protein of interest on a filter after running it on a gel. And again, this would be the confirmatory test for HIV. Choice C is a PCR reaction, which you could actually use to measure the viral load of HIV. And then D and E are Southern and Northern blots, which you can use a DNA probe to detect DNA or RNA.